Good day, this is Professor Will, CBMD PhD. Today is Thursday, June 11, 2020. It is 7:02 and 5 seconds a.m. Eastern Standard Time. And today I'm going to be doing a research study on bioinformatics. So let's get started here. Information retrieval from biological databases. Tissue type. Along with the scientific name is the information and taxonomic classification of the organism. Different levels of the classification are hyperlinked to the NCBI taxonomy database with more detailed description. This is followed by the reference field, which provides a publication citation related to the sequence entry. The reference part includes author and title information of the published work, or tentative title for unpublished work. The citation is often hyperlinked to the PubMed record for access to the original literature information. The features section includes annotation information about the gene and gene product, as well as regions of biological significance reported in the sequence with identifiers and qualifiers. The source field provides the length of the sequence, the scientific name of the organism, and the taxonomy identification number. Some optional information includes the clone source, the tissue type, and the cell line. The gene field is the information about the nucleotide coding sequence and its name. For DNA entries, there is a CDS field, which is information about the boundaries of the sequence that can be translated into amino acids. For eukaryotic DNA, this field also contains information of the locations of exons, and translated protein sequences is entered. The third section of the flat file is the sequence itself, starting with the label origin. The format of the sequence display can be changed by choosing options at a display pull-down menu at the upper left corner. For DNA entries, there is a base count report that includes the numbers of A, G, C, and T in the sequence. This section for both the DNA or protein sequences ends with the two forward slashes symbol. In retrieving DNA or protein sequences from GeneBank, the search can be limited to different fields of annotation such as organism, accession number, authors, and publication date. One can use a combination of limits and preview index options as described. Alternatively, a number of search qualifiers can be used, each defining one of the fields in a GeneBank file. The qualifiers are similar to but not the same as the field tags in PubMed. For example, in GeneBank, gene represents field for gene name, auth for author name, and org for organism name. Frequently used GeneBank qualifiers, which have to be in uppercase and in brackets. Alternative sequence formats. FASTA. In addition to the GeneBank format, there are many other sequence formats. FASTA is one of the simplest and most popular sequence formats because it contains plain sequence information that is readable by many bioinformatics analysis programs. It has a single definition line that begins with a right angle bracket, followed by a sequence name. Sometimes extra information such as GI number or comments can be given, which are separated from the sequence name by A and bar symbol. The extra information is considered optional and is ignored by sequence analysis program. The plain sequence and the standard one-letter symbol starts in the second line. Each line of the sequence data is limited to 60 to 80 characters in width. The drawback of this format is that much annotation information is lost. Abstract Syntax Notation 1, ASN Decimal 1, is a data markup language with a structure specifically designed for accessing relational databases. It describes sequences with each item of information in the sequence record separated by tags, so that each subportion of the sequence record can be easily added to relational tables and later extracted. But more difficult for people to read, this format makes it easy for computers to filter and parse the data. This format also facilitates the transmission and integration of data between databases. Conversion of sequence formats. In sequence analysis and phylogenetic analysis, there is a frequent need to convert between sequence format. One of the most popular computer programs for sequence format conversion is ReadSeq. It recognizes sequences in almost any format and writes a new file in an alternative format. It also offers direct access to certain sequence analysis applications such as sequence similarity searching and clustral sequence alignment. Queries can be launched using quick text search with only one query box in which to enter information. There are also more elaborate submission forms, the standard query form and the extended query form. The standard form allows for criteria fields to be used, which are linked to Boolean operators. The extended form allows many more diversified criteria and fields to be used. The search results contain the query sequence and sequence annotation, as well as links to literature, metabolic pathways, and other biological databases. Summary. Databases are fundamental to modern biological research, especially to genomic studies. The goal of a biological database is to fold information retrieval and knowledge discovery. Electronic databases can be constructed either as flat files, relational, or object-oriented. Flat files are simple text files and lack any form of organization to facilitate information retrieval by computers. Relational databases organize data as tables and search information among tables with standard features. Object-oriented databases organize data as objects and associate the objects according to hierarchical relationships. Biological databases encompass all three types. Based on their content, biological databases are divided into primary, secondary, and specialized databases. Primary databases simply archive sequence or structure information. Secondary databases include further analysis on the sequences or structure. Specialized databases cater to a particular research interest. Biological databases need to be interconnected so that entries in one database can be cross-linked to related entries in another database. Effective information retrieval involves the use of Boolean operators. NCBI databases accessible through entries are among the most integrated databases. Alternatively, one can use NCBI specific field qualifiers to conduct searching. To retrieve sequence information from NCBI GeneBank, an understanding of the format of GeneBank sequence files is necessary. It is also important to bear in mind that sequence database in these databases are less than perfect. There are sequence and annotation errors. Biological databases are also plagued by redundancy problems. There are various solutions to correct annotation and reduce redundancy. For example, merging redundant sequences into a single entry or store highly redundant sequences into a separate database. Sequence comparison lies at the heart of bioinformatics analysis. It is an important first step towards structural and functional analysis of newly determined sequences. As new biological sequences are being generated at exponential rates, sequence comparison is becoming increasingly important to draw functional and evolutionary inference of a new protein with proteins already existing in the database. The most fundamental process in this type of comparison is sequence alignment. This is the process by which sequence is compared by searching for common character patterns and establishing residue-residue correspondence among related sequences. Pairwise sequence alignment is the process of aligning two sequences. It is the basis of database similarity searching and multiple sequence alignment. Evolutionary bases. DNA and proteins are products of evolution. The building blocks of these biological macromolecules, nucleotide bases, and amino acids form linear sequences that determine the primary structure of the molecule. These molecules can be considered molecular fossils that encode the history of millions of years of evolution. During this time period, the molecular sequences undergo random changes, some of which are selected during the process of evolution. As the selected sequences gradually accumulate mutations and diverge over time, traces of evolution may still remain in certain portions of the sequences to allow identification of the common ancestry. The presence of evolutionary traces is because some of the residues that perform key functional and structural roles tend to be preserved by natural selection. Other residues that may be less crucial for structure and function tend to mutate more frequently. For example, active site residues of an enzyme family tend to be conserved because they are responsible for catalytic function. Therefore, by comparing sequences through alignment, patterns of conservation and variation can be identified. The degree of sequence conservation in the alignment reveals evolutionary relatedness of different sequences, whereas the variation between sequences reflects the changes that have occurred during evolution in the form of substitutions, insertions, and deletions. Identifying the evolutionary relationships between sequences to help characterize the function of unknown sequences. When a sequence alignment reveals significant similarity among a group of sequences, they can be considered as belonging to the same family. If one member within the family has a known structure and function, then that information can be transferred to those that have not yet been experimentally characterized. Therefore, sequence alignment can be used as basis for prediction of structure and function of uncharacterized sequences. Sequence alignment provides inference for the relatedness of two sequences under study. If the two sequences share significant similarity, it is extremely unlikely that the extensive similarity between the two sequences has been acquired randomly, meaning that the two sequences must have derived from a common evolutionary origin. When a sequence alignment is generated correctly, it reflects the evolutionary relationship of the two sequences. Regions that are aligned but not identical represent residue substitution. Regions where residues from one sequence corresponding to nothing and the other represent insertions or deletions that have taken place on one of the sequences during evolution. It is also possible that two sequences have derived from a common ancestor, but may have diverged to such an extent that the common ancestral relationships are not recognizable at the sequence level. In that case, the distinct evolutionary relationships have to be detected using other methods. Good day, this is Professor Will, CBMD PhD on microbiology, bioinformatics, and pathophysiology. Well, hope you enjoyed listening. Until next time, have a great day.